Hello everyone, welcome to lesson 1.2. And over the course of these two lessons, we're going to be going over and offering a basic introduction to the two distinct, unique cells that all living organisms possess, that being prokaryotic cells and eukaryotic cells. So we're going to discuss structures and functions that encompass both of these cells and other basic information regarding both of the types of cells. So without further ado, let's get into it, starting with the prokaryotes or prokaryotic cells. So before going into prokaryotic cells, it's very important to discuss and offer an introduction to a certain tool that we use called electron microscopes in biology. And electron microscopes are very important and essential in biology as they allow us to see things as small as 0.001 micrometers. And they also have a much higher range of magnification and higher resolution in terms of actually magnifying and increasing the quality of certain images when looked under the microscope. So they're a very important tool in higher level biology. They can also reveal the ultrastructure of cells, including ribosomes, lysosomes, the endoplasmic reticulum. They allow us to see dark green areas in chloroplasts called grana, and they also allow us to see the internal membranes of the mitochondria. And here are the just two images that really distinguish the quality between a light and electron microscope. So on the left, as you can see here, this would be image of a honeybee and an ant under a light microscope. And on the right, this would be the same image, a honeybee and an ant under, or the same specimen under an electron microscope. You can really see the differences that are highlighted and emphasized between the two types of microscopes. All right, now moving on to prokaryotic cells. So there are two main types of organisms, and they are grouped together according to their cell type. And these two cell types are eukaryotic cells and prokaryotic cells. So eukaryotic cells contain a nucleus that holds the DNA within the actual cell, and prokaryotic cells do not contain a nucleus, and their DNA is free floating within the cytoplasm. That's really how you distinguish both of these cells from each other. Prokaryotes. So, prokaryote cells are single celled organisms that lack membrane bound nuclei and organelles within them. They are thought of as the first organisms to have ever evolved on Earth, and this is due to the fact that they have the simplest and most primitive biological structures out of the two types of cells. And prokaryotes are also the smallest organism on Earth, and most are between 1 micrometer to 2 micrometers. All right, so moving on to structures and functions of prokaryotic cells. So prokaryotic cells contain a cell wall, and this offers structural support and protection. They also contain a plasma membrane, which controls entry and exit of substances within the cell, and it pumps some of them in by active transport. And this is very similar to eukaryotic cells. Prokaryotic cells also contain cytoplasm throughout their entire cell, and it contains enzymes that's needed for all metabolic processes in cells for cellular respiration and other reactions. Prokaryotic cells also contain ribosomes. However, the distinction between eukaryotic cells is that prokaryotic cells contain 70 size ribosomes, not 80. And all ribosomes in prokaryotic cells are free in the cytoplasm, and they're used for protein synthesis. All right, naked DNA. So, naked DNA is the genetic material and information that's stored inside the prokaryotic cell. It is stored in the nucleoid, which is free floating in the cytoplasm of the cell. It's not surrounded by a nuclear membrane. It's circular in shape, and it is not associated with proteins, which is why it is referred to as naked, because it's not associated with any type of protein coiling. All right, moving on to the pili. So the pili are thread-like projections that are usually more numerous in abundance than flagella. They're not usually involved in movement and instead allow bacteria to stick to other cells, other bacteria, and they're involved in the transferring of genetic DNA and information to other cells via a process called conjugation. This can really help in genetic variability and diversity within certain populations of bacteria. And finally, we have the flagella, and this is usually a long, thin, whip-like structure that's used for movement and locomotion of a prokaryotic cell. And here's just a very brief diagram that really emphasizes all of the structures of a prokaryotic cell. You have your cell wall here, and that is made of peptidoglycan. You have the plasma membrane, or the cell membrane on the inner area, this turquoise color. Then you have your cytoplasm all throughout the cell, your free-floating 70 size ribosomes, these little dots. The pili, the thread-like projections, high in abundance, projecting off of the cell, actual prokaryotic cell. You have your nucleoid, which contains the naked DNA. And finally, you have your flagella right here, this pink structure. And here is just a quick diagram that really emphasizes what you need to focus on when you're actually drawing and illustrating scientific illustrations regarding prokaryotic cells. So it's just sort of a quick checklist just to check off to ensure that you're including everything, including the pili, cytoplasm, the nucleoid, the cell wall. Make sure that in brackets it's written as peptidoglycan, a cell membrane, the plasmids, the flagellum right here, and the 70 size ribosomes. All right, prokaryotic cell division. So all living cells need to produce new cells, and they come from pre-existing cells, as we know from the cell theory. Prokaryotic cells, however, do divide through a unique fashion as opposed to eukaryotic cells. Whereas eukaryotic cells undergo mitosis to divide, prokaryotic cells actually undergo binary fission. And this is a form of asexual reproduction. Binary fission happens very quickly and and rapidly, and the end product is two genetically identical daughter cells. So as you can see right here in the following image on the screen, it really just emphasizes the speed and manner in which binary fission actually occurs in various prokaryotic cells. So during binary fission, a parent cell will divide into two genetically identical daughter cells. And each daughter cell will receive an exact and complete copy of the genetic material and DNA from the actual parent cell. And any deviations or alterations from this parent DNA would be a mutation. And since bacteria reproduce very rapidly, there is an increased rate of mutations during binary fission, and thus this results in an increased genetic diversity within populations of prokaryotic cells. All right, so moving on to reproduction in bacteria and other living organisms. So as we know before from the seven functions of life, all organisms have to reproduce, and they can either reproduce through asexual means or sexual means. So during asexual reproduction, a single individual or organism is the sole parent, and the single parent will pass all of its genes and DNA off to its offspring. And the offspring are thus genetically identical to the parents. And this will result in genetic clones, and they're genetically identical to the actual parent. And any deviations as a result will be the result of genetic mutations. During sexual reproduction, two parents will give rise to an offspring together. Each parent will pass on half of its genes to the offspring, and thus the offspring will have a unique combination of both genes inherited from both parents. And this results in greater genetic variation because offspring can vary genetically from siblings and parents as a result. Alright, so moving on to negative effects of prokaryotes. So the primary negative effect that prokaryotic cells have is that they are responsible for many diseases. And this is due to the fact that infectious bacteria and other disease-causing agents, such as viruses and microorganisms, are primarily composed of prokaryotic cells or bacteria. And prokaryotic cells and bacteria can also infect livestock and crops and therefore threaten our primary food source as humans. Now, of course, there actually are some positive effects that prokaryotes can have on different ecosystems and environments and biological systems. For one, have very important and essential influences and impacts on ecosystems, as they act as decomposers and they can help cycle nutrients throughout ecosystems. Bacteria and prokaryotic cells can also reside in the intestines of animals and aid and assist in digestion. Bacteria and prokaryotic cells can additionally be used for commercial use in the making and manufacturing of yogurt, cheese, and chocolate. And finally, prokaryotic cells and bacteria can also be used to produce antibiotics. And that's going to be it for the lesson on prokaryotic cells. I'll see you in the next lesson covering eukaryotic cells. Hello everyone, welcome to the second part of lesson 1.2. In this portion of the lesson, we're just going to be doing an extension similar to the first part, in which we're just going to be going over a basic introduction to eukaryotes and their structures and functions and other information along those lines. So let's get into it. Eukaryotic cells. So eukaryotic cells are thought of to have more complicated and complex internal structures and physiology in comparison to prokaryotic cells. This is due to the fact that eukaryotic cells are compartmentalized, and all of the organelles in eukaryotic cells are also membrane bound. So and all of the organelles within eukaryotic cells, similar to prokaryotic cells, have distinct structures and functions. Here are just some four advantages of having membrane-bound organelles in an actual cell. And this is that enzymes and substrates for a particular process can be more concentrated than if they were spread throughout the cytoplasm. They're contained within a membrane-bound organelle. Substances that could cause damage to the cell can be kept inside the membrane of an organelle. Conditions such as pH can be maintained at an ideal level for a particular process. And organelles with their contents can be moved around the cell more conveniently and easily if they're confined within membranes. So moving on to drawing the ultrastructure of eukaryotic cells based on electron microscopes. So whenever you're drawing or illustrating scientific illustrations of actual eukaryotic cells and labeling their organelles, you always want to ensure that your drawing includes the following organelles and structures, including the plasma membrane, the nucleus, the rough endoplasmic reticulum, the Golgi apparatus, the lysosomes, mitochondria, free ribosomes at 80 size, chloroplasts if it's a plant cell, vacuoles and vesicles, microtubules and centrioles, cilia and flagella, and a cell wall if it is a plant cell. And here is just a very basic drawing of a eukaryotic cell. So as you can see, certain elements of the drawing are the lysosomes right here, the mitochondria over here, rough endoplasmic reticulum here with ribosomes spread out and distributed throughout all of them, the smooth endoplasmic reticulum that lacks any and all ribosomes, the cytoplasm throughout the entire cell, the nucleus, this circular center organelle would be the nucleus, Golgi apparatus over here, and then free ribosomes that are 80 size within the actual entire cell, and then the plasma membrane, which is this outer area. All right, so now moving on to the specific organelles and their functions. So first we have the plasma membrane. And the plasma membrane's main function is to control and mediate what enters and leaves the cell. And the membrane is a double layer of water repellent molecules that enable certain molecules to enter it and prevent other molecules from entering it that could cause damage to the actual cell. All right, moving on to the nucleus. So the nuclear membrane has a double membrane with pores, and these pores contain chromosomes. And the chromosomes is essentially just DNA that's associated with proteins. The uncoiled chromosomes in the nucleus are called chromatin, and these are what the densely stained regions around the edge of the nucleus are, the chromatin. And the function of the nucleus is that it is the site of DNA replication in the cell and the transcription to mRNA. And then RNA is exported from the nucleus via the nuclear pores to the cytoplasm of the cell. Rough endoplasmic reticulum. So the rough endoplasmic reticulum consists of a flattened membrane sacs that are called cisternae, and attached to the cisternae are 80 size ribosomes, and they are distributed pretty much evenly throughout the entire rough endoplasmic reticulum. The function of this organelle is for protein synthesis, and this is due to the fact that proteins are made on the ribosomes of the rough endoplasmic reticulum. They're passed into the cisternae, and they're carried off by vesicles which bud off and are moved to the Golgi apparatus. All right, Golgi apparatus. So this organelle consists of flattened membrane sacs called cisternae as well. The function of this organelle is to process the proteins that are brought in by vesicles from the rough endoplasmic reticulum. The proteins are then carried in the vesicles to the plasma membrane for secretion from the cell. Lysosome. So these are organelles that are formed by the Golgi apparatus. Their primary function is to digest old organelles, engulf bacteria and viruses. They can also break down biomolecules, including proteins, nucleic acids, carbohydrates, lipids, and cell debris in the cell. Mitochondria. So mitochondria are surrounded by a double membrane, and then the inner membrane has folds that are called cristae. And there's a fluid inside the mitochondria that is called the matrix. And the primary function of the mitochondria is to produce ATP for the cell by aerobic cellular respiration. Free ribosomes. So the distinction is that ribosomes within eukaryotic cell are all 80 size. These free ones are not surrounded by a membrane, and they instead are free floating within the cytoplasm, and their primary function is to make and synthesize proteins and release them into the cytoplasm. Chloroplast. So these are plant exclusive organelles only. They have double membranes and they're only found in plants. They are the site of photosynthesis in plants. This is because light is trapped within the chloroplast by chlorophyll. And then carbohydrates are made, using the trapped energy and carbon dioxide in the stroma of the chloroplast. And then oxygen is produced as a waste gas from the actual process of photosynthesis. Vacuoles and vesicles. So these are single membrane organelles that contain fluid inside of them. Most plants typically have a large central vacuole, and the function of the vacuole is that it is mostly used for the storage of amino acids and sugars. However, some animals will digest food inside of vacuoles. And then vesicles are found both in plant cells and animal cells, and their function is to transport materials throughout the entire cell through endocytosis and exocytosis. All right, and finally, cilia and flagella, and these are exclusive to animal cells. And as we know before, flagella is a whip-like structure that projects from the cell surface, and they are usually larger and usually only one is present for locomotion, whereas cilia are smaller and many are present. So they can be found in stomach cells and respiratory cells as well. However, for both organelles, their primary function is for locomotion and movement. And here is just a abbreviation to help you remember the differences between the prokaryotic cell and a eukaryotic cell through the abbreviation DORA. So for prokaryotes, for their DNA is naked, it's circular in shape, and there are usually no introns. For eukaryotes, their DNA is bound to a protein, it's linear in shape, and it usually has introns. For organelles, prokaryotes contain no or possess no nucleus. They have no membrane-bound organelles, and they have 70 size ribosomes. Eukaryotes have a nucleus and possess one. They possess membrane-bound organelles, and they also have 80 size ribosomes instead of 70. For reproduction, prokaryotic cells divide by binary fission, and they are single chromosome. They have single chromosomes, so they're haploid. For eukaryotes, they divide by mitosis and meiosis, and their chromosomes are paired, or they're diploid. And for the average size, prokaryotes are much smaller in comparison, around 1 to 5 micrometers, and eukaryotes are much larger, around 10 to 100 micrometers. So that's it for lesson 1.2, covering prokaryotes and eukaryotes. Thank you for listening, and I'll see you next time.